Hello everyone, I am Rajiv Saxena, Associate Professor, Smriti College of Pharmaceutical Education, Indore. In today's topic, we'll discuss about bacterial growth cycle and we'll also discuss about different categories of the culture media. So our first topic is bacterial growth cycle or bacterial growth curve. In the last class, we have discussed about the growth and growth requirements of a bacteria. So again we'll revise two things. Number one, if we are talking about the growth of a bacteria, we are actually talking about increase in the number of the bacteria, and for the proper growth of a bacteria, some nutritional requirement and some physical growth requirements are necessary. And if we provide those requirements at fullest, then definitely our bacteria will grow at the optimum rate. So again, if we are talking about some industrial microorganism or industrial bacteria, the first task for the industry is we have to go for the optimization of various nutritional requirement and the physical requirement. We cannot say like every bacteria or every microorganism have the similar kind of the requirement. Whatever the bacteria that is with us, that is provided by the industry, first we have to go for the optimization study of the nutritional factor also and of the physical factor also. So every bacteria, say for example, if we are having two bacteria, one is A and one is B, then both the bacteria may have the different requirement of nutrition and different requirement of the physical. Okay, like for example, temperature requirement may differ, pH requirement may differ, the concentration of carbohydrate, the concentration of carbon, the concentration of nitrogen that is required by a particular bacteria may have different tendency as compared to that of the other bacteria. So, once we go for the optimization of the nutritional requirement and the physical growth requirement, the next task is the bacterial growth curve study. Now, for example, if we are having some bacteria which is dormant in nature and we are introducing that bacteria in an optimized media, whatever the media conditions we have optimized, whatever the physical growth conditions that we have optimized, and if a new bacteria or a dormant bacteria is introduced in that new environment, then it is not like so that the bacteria, as soon as it will be introduced in the growth environment, readily it will start growing. This is not the condition. Like when a new bacteria or when a dormant bacteria is introduced into a new media or a new nutritional environment, first it has to undergo different phases and then it will reach to a point where the actually visible growth of the bacteria is observed. So in order to understand the behavior of a bacteria in a new culture media, then first of all we have to study the bacterial growth curve, and most of the bacteria follow the similar growth curve in their cycle. So the bacterial growth curve or the bacterial growth cycle is actually divided into four phases. Number one is a lag phase. Number two is a exponential phase, or it is also called as a log phase. Number 3 is a stationary phase, and number 4 is a decline phase. Again, I am repeating the different phases. The bacterial growth curve or the bacterial growth cycle is actually divided into 4 phases. Number 1 is a lag phase. Number 2 is a exponential growth phase. Yeah, it is also called as a log phase. The third phase is the stationary phase, and number 4 is a decline phase. So one by one we will study what actually happens to a bacteria when it crosses these phases of the growth cycle. The first phase is the lag phase. Now we have to understand in such a manner, like when a new bacteria or a dormant bacteria is introduced into a new environment, definitely it has to adjust itself with the new environment. It has to go for the production of various metabolites. It has to go for the production of various macromolecules which are necessary for the growth. So this is because, in other words, we can say that this lag phase is actually the adaptation time when a bacteria adapt itself with the presence of the external environment in terms of the nutrition also and in terms of the physical requirement also. So it has been observed, like when your bacteria is in the lag phase, then you will see that there is no particular growth or a visible growth of a bacteria can be seen, because this is actually the time period when bacteria is preparing itself so that in the next phase it can go for the multiplication or for the exponential growth. So we can say that this is actually the preparatory phase. When a bacteria is preparing itself, it is synthesizing all the essential nutrients. It is go for adjusting with the different macromolecules which are available in the external environment, and it is adopting itself in a particular way so that it can go for the multiplication in the next phase. Now, once the lag phase is completed, the bacteria enters into the next phase, which is called as the exponential phase. It is also called as logarithmic phase, or it is exactly the phase where you can see the visible growth, visible increase in the number of bacteria, or the multiplication of the bacteria actually occurs in the exponential phase. Since the bacteria is already adopted in the lag phase, now it is having all the essential elements surrounding the cell which is required for the growth, and all the physical requirement that we have already fulfilled and all the nutritional requirements are already fulfilled. So the bacteria will undergoes the multiplication in the exponential growth phase. Now, the exponential growth phase in terms of a pharmaceutical industry or any industry which is utilizing the bacteria for the production of different products is very important, because the task of every industry is like the exponential phase should be very high, because it is actually the phase where the production of different categories of the pharmaceutical, like for example, if you are talking about the recombinant product, if you are talking about the vaccines or antibiotics, then the maximum growth or the maximum production of these particular product occurs in the exponential phase. These products are not generated in the lag phase, but it is exactly generated in the exponential growth phase. So if in terms of industry, if we are talking about in terms of industry, then definitely our target is that we have to go for the optimization study of the nutrition and the physical requirement, so that the exponential growth phase can have the longer duration, because if the duration is longer, then definitely more amount of product will be generated. So from industry point of view, this exponential phase or the log phase is very essential. Now, the third phase of bacterial growth cycle is the stationary phase. What happens when a bacteria is multiplying? It is consuming all the nutrients which are available in the surrounding. But a time will come when the nutritional quality of the environment reduces, and also the generation of various toxic material increases. And apart from that, many of the cell after the growth, they will undergoes the death phase. So a time will come when the growth of a bacteria and the death of the bacteria, they are balanced. So when these type of balances are observed, then these type of phases are called as the stationary phase of the bacteria. Because once the logarithmic phase is over, then we will find that our product is generated. But apart from the generation of the product, the nutrients which are available in the environment, they are also reducing. The quality of the nutrients are also reducing, and various toxic metabolites are generated by the bacteria. So these toxic element and the various death that occurs of the bacteria actually somehow maintains the balance between the growth and the death of the bacteria. So there is a stationary phase in which the growth of the bacteria and the death of the bacteria, it is somehow balanced. Then that particular phase of the bacterial growth cycle is called as the stationary phase. So till now we have studied three phases. Number one is the lag phase, in which the bacteria is adjusting itself according to the environment, and once the adjustment is over, then it will undergoes the multiplication or the logarithmic increase in the number of the bacteria, where the metabolic cycles inside the bacteria grow at a very faster rate, and different metabolites, different products are generated in the exponential growth phase. But when the growth of a bacteria and the death of the bacteria, it is balanced, then that phase is actually called as a stationary phase. Now, once the stationary phase is over, then the next bacterial growth curve indicates the formation of the decline phase. Now, what happens in the decline phase? When the concentration of the toxic element is very high, and when the death supersedes the growth of the bacteria, then definitely it is observed that the number of bacteria, they are declining and the growth is less, and the death is more. The generation of the toxic element is very high and the metabolic phases or metabolic cycle inside the bacteria is also reducing, and the quality of the nutrients which is available in the environment is also reducing to a very lesser extent. So it is definitely the phase in which the metabolic activity is very less and the death of the bacteria is superseding the growth. So that's why this phase is actually called as the decline phase of the bacteria. The next topic of today's session is culture medias. Like in the last class, we have studied about the bacterial growth curve, growth requirements, including the nutritional requirement and the physical growth requirement. But once we have come into the conclusion that for a particular bacteria, this nutrition is required. So when we are cultivating a bacteria, when we are in the phase of growing a bacteria, definitely some preparation is required in which your desirable bacteria can be inoculated. So the preparation that favors the growth of your bacteria is actually called as the culture media. It is having all the necessary ingredients which are required for the growth of that particular bacteria. So these culture medias are again divided into different types. But broadly we can say that these culture medias are of four types. Number one is called as the chemically defined media. Second one is a complex media. Number three is a selective or differential media. And number four is a enrichment media. So one by one we'll discuss all these four different kinds of the culture media. Number one is a chemically defined media. As the name itself is suggesting, it is the type of a media in which the chemical composition of all the individual ingredients is well known. Here I have given one example, like for example for a particular bacteria, I have optimized one recipe or I have optimized one preparation that is helpful in the growth of that bacteria. But in this recipe or in this preparation, whatever the ingredients I am adding, the exact chemical composition of individual ingredient is well known. For example, glucose 5 grams, ammonium phosphate 1 gram, sodium chloride 5 gram. So all the ingredients which are utilized for the preparation of this recipe, they contain the composition, the exact amount of every ingredient that is contributing in the growth of the bacteria is very much defined. So these type of culture medias are called as chemically defined media. The next culture media is a complex media. Now complex media, in contrast to the chemically defined media, is a type of media. Definition wise we can say that it is a type of a media in which the exact chemical composition is not defined. Now how we can understand this? Like I have given one example, like in this recipe that we have optimized for a particular bacteria, we have added peptone 5 gram, beef extract 3 gram, but it is peptone and beef extract, they are very rich source of different categories of the nutrition. Like beef extract can offer the nitrogen sources also, it can offer the carbon sources also, peptone is again a very good source for the nitrogen, but the composition, the chemical constituent in terms of number is not defined in this type of a preparation. So that's why we are calling it is a complex media in which the exact chemical composition of an individual ingredient is not defined. Definitely, like for example, if I am talking about a peptone or a beef extract, they are very nutritious in nature. It is having all the components like carbohydrate, proteins, lipids, all the essential ingredients are available in the beef extract and the peptone. But in that extract or in that composition, the individual component is not very much defined. So these type of a culture media is called as a complex media. Now, the third category of the culture media is a selective media or a differential media. Now, first we'll understand what is the meaning of the selective media or the differential media. Okay. So, for example, in a laboratory or in an industry or in any R&D unit, if we are working on a particular bacteria, say for example A bacteria, so our intention in that case will be like that whatever the culture media that we are using, it should favor the growth of our desirable bacteria. It should not favor the growth of all the different categories of the bacteria. Because what will happen if you are preparing a culture media, it is, for example, giving the favor to the growth of your desirable bacteria. But apart from that, it is also favoring the growth of n different categories of the bacteria. Then what will happen? The nutrition will be shared by undesirable bacteria and the desirable bacteria. So whatever the nutrition we have planned for the growth of your desirable bacteria will again be shared between the undesirable bacteria. So definitely the growth of the undesirable bacteria may be more as compared to that of the desirable bacteria. So in many cases, even in various pathology laboratories, it is required that whatever the culture media that we are preparing, it should have the element that is favoring the growth of any particular category of a bacteria. So for that, in that case, we have to go for the use of the selective media or the differential media. The selective media will do what? It will selectively go for the growth of your desirable bacteria, and a differential media will be a kind of a media that may favors the growth of second bacteria or third bacteria also, but you can easily visualize or you can easily go for the isolation of your category, your desirable category of the bacteria. So differential media can differentiate between the desirable and the undesirable bacteria, whereas the selective media is a kind of a culture media that selectively favors the growth of your desirable bacteria. Some examples are given like bismuth sulfate agar media. It is very selective media for the growth of the Salmonella typhi. Some more examples are there like brilliant green agar media. It is for gram-negative Salmonella. Mannitol salt agar media. It is for S. aureus. Blood agar media. It is for Streptococcus pyogenes. So these are the different categories of the culture media that selectively grow your desirable category of the bacteria, or the bacteria that is under investigation, the bacteria that you are using, or the bacteria that is utilized for the industrial production of a particular microorganism. Now, coming to the last category of the culture media, it is called as a enrichment media. Now, enrichment media is somewhat similar to that of the selective media. So, we can contrast the definitions of the selective media and the enrichment media. So, what selective media is doing? Selective media is actually selectively growing particular category of a bacteria. Whereas if you are talking about the enrichment media, these are the media which are generally utilized when the concentration or when the amount of your bacteria which is under investigation or you are studying, if that amount is very less, then what the enrichment media will do, it will suppresses the growth of other category of the bacteria, whereas it will grow your desirable category of the bacteria. So suppression of the undesirable bacteria and the growth of your desirable bacteria is actually the main purpose of using the enrichment media. So these are the different categories of the culture media which are routinely used in any microbiological laboratory or in any industrial production houses for the growth of different microorganisms. Especially in this case, we are talking about the bacteria. But again, we can say that the culture medias, in one more way, it can be classified that these culture medias can either be a solid media or it will be a liquid media. Like for example, solid agar media. If in the media, if we are adding a agar, then agar is a solidifying agent, and because of the presence of the agar, the media will get solidified. But for various routine microbiological experiment, sometimes the liquid media is required. Like whatever the compositions we are preparing, we are adding all the components and we are adding the water, but we are not adding any solidifying agent like agar. Then that media will be in the form of the liquid condition. So that particular liquid media is actually called as a, in terms of microbiology, it is called as a culture broth. So basically we can divide the culture media into 4 types. Number 1 is a chemically defined media. Number 2 is a complex media. Number 3 is a selective or differential media. Number 4 is a enrichment media. But depending upon the consistency, depending upon the state of the culture media, your culture media can be a solid media or it can be a liquid media, and liquid media in terms of microbiology it is called as a broth. So if you are not adding a solidifying agent, then it will be in the form of a broth, and if you are adding a solidifying agent, then it will be in the form of a solid condition.